Good morning. My name is Angélica Isa, and today I will be presenting on behalf of my colleagues Carolina Parada, Marilyn Palomino, and Eliana Quispitupac. I wanted to begin this presentation with a brief introduction to the museum and archaeological site where the studied beetle derives from. The Pachacamac Archaeological Sanctuary was the home of the most important oracle of the central coast of Peru during the pre-Hispanic period, and can boast an almost continuous occupation for over 1,200 years. The first interpretation space or site museum in Pachacamac was built in 1965. This is an archaeological gourd which was registered in 2013 before I started working at Pachacamac. You can see it had active damage when it was registered, but there being no conservators on the team for the non-textile collections at the time and a limited understanding of insect damage, it was not acted upon at the time and the gourd was stored. Fast forward to 2014, and after many years of planning, the original 1965 museum was torn down in order to build a new museum on the same grounds. So as you can imagine, this involved a couple years of organizing, checking, packing, moving, rehousing. I began work at the museum officially in 2016, just as the new museum was being handed over by the construction company. And after a few months of settling in, because I had to help with the setup of the new museum, I began an exhaustive check of all collections. I finally got around to the gourds, and this is what I found in August 2017. At that point, we had not yet identified the beetle, which was associated to the destroyed gourd, but I did learn to recognize it. And although I'm not showing the whole photo here, because these are human remains, this is the body of a woman I like to call Snow White, because at some point in the past, she was set on a bed of sand, surrounded by associated offerings, both organic and inorganic. You can see an archaeological corn cob offering, and she was encased in a glass box for display. So you can see here that when I found her in 2017, there were piles of the unknown beetles surrounding all the edges of the glass box. In the same quarantine room where she was being provisionally stored, we installed the chest freezer, and you can see that some of these beetles were attracted to the moisture around the edges of the freezer seal. This is another image of the museum during the pandemic last year. We were gone for months, starting in March 2020, under severe lockdown, and we were able to do only a few quick visits. The museum is 30 kilometers south of Lima, so it's not that easy to get there. You can see the sheer amounts of these insects as I pick them up in that little pot. So a little background information. In Peru, all woodworm tends to be colloquially described as polilla or moth, and we use the term apolillado or mothed to mean objects are being destroyed by insects or that have been destroyed by insects. These objects can be wood, clothes, even food. So the misnomer moth tends to be applied to everything from actual moths to termites to beetles. The beetle shown here is the main cause for organic destruction in the Pachacamac collections, and we didn't know what it was. We didn't have entomologists at the time, and the published glossaries that I had access to tend to apply to more temperate areas. We could not find this beetle in them. The fumigations, which are regularly carried out in museums here as part of contractual obligations with the cleaning companies, did not affect the beetle at all. So we came up with the goals for this study. One of the goals was to identify the main beetle, then create a Peru-specific glossary and experiment with the possibility of harnessing traditional knowledge to create a safe and economic way to repel all these damaging beetles. So, because I had pictures of these beetles, we had an initial potential ID, thanks to Dr. Tony Irwin and the Pest List group, as Trichorinus herbarius, or the Mexican book beetle. We assembled a team which included entomologists, chemical engineers, and a conservator, and myself as a representative for the museum, and we collected the specimens from infected organic archaeological material. And you can see we actually did this during the pandemic last year, during one of these few visits that we were allowed to do. For mass rearing, the larvae were fed with dried fragments of the same gourds that are found in archaeological and historical objects in museums, which is Lagenaria ciceraria by Malin and Standel. And the new generation that emerged from this base group was used for the chemical tests. The entomologist in our team is doing a master's dissertation on this. She extracted the male genitalia to finalize the identification, and she ran all the keys. And while we are still awaiting a peer-reviewed confirmation for her ID, we are quite sure that Dr. Irwin was correct, and we are talking about the Mexican book beetle. So our thesis student, Marilyn Palomino, has done her biological study, which she's still carrying out at the moment. And the findings regarding the beta biology so far can be summarized as follows. The entire cycle from egg to adult in artificial rearing conditions is approximately 146 days. After copulation, the gravid females place their eggs one by one among the folds of the substrate, and the eggs are not adhered to the surfaces in any way, which causes them to move and roll around easily over the surface. Incubation lasts around 16 days, after which actively feeding larvae spend about 102 days or so creating internal galleries in the substrate and preparing an inner chamber where they will spend about 25 days as a pupa until the emergence of a completely sexually mature adult. So the second part of this study is the tarwi, right? So we identified the beetle, and then we looked at the tarwi. This plant is endemic to the Andean region, and it has been used by Peruvian rural communities since pre-Hispanic times for control of agricultural pests and as a natural antiparasitic in animals. So it is common to see it planted around crops in order to protect them. Tarwi produces quinolizidinic alkaloids, I hope I didn't mess up that word, and other metabolites which are distributed in its leaves, flowers, and seeds. Now, although the role of alkaloids can still be debated, research suggests that among other roles, they protect plants against both herbivores and pathogenic microorganisms. And if you're not familiar with tarwi, if you've never heard of it yet, you might become familiar with it over the next few years because tarwi is known to have over 40% protein content. And I really believe it has the potential to become the next quinoa in Western markets. So we were aware of the work carried out with neem and citronella abroad, but we don't have those plants here. So we propose the use of alkaloids extracted from tarwi to repel the Trichorinus herbarius and avoid the use of toxic substances which could affect museum staff and organic collections. So how did we get this? Thanks to the collaboration of Tarwi Corp, is a local company which processes tarwi seeds for human consumption in the same way that we eat beans or quinoa or lentils, right? We obtain tarwi leaves and seeds from them. From these, we applied a process to obtain a hydroalcoholic extract, which is rather complicated, which you can check in the bibliography for details if you want to reproduce this study. So I'm only going to summarize the process here. So for the leaves, we ground them, macerated them in ethanol for 3 days, filtered and stored the liquid. And for the seeds, we dried them in an oven at 40 degrees Celsius for 2 days, ground and pulverized them in a food processor, and then macerated some of the ground seeds in ethanol for a few days, and finally filtered and stored as well. So that's how we made the 2 hydroalcoholic extracts. Because tarwi is generally extremely bitter, it needs to be washed several times before human consumption. So Tarwi Corp does this washing, and we obtain fresh aqueous effluents from the washing processes from them. This is wastewater that they just throw out most of the time because there's nothing they can do with it. It's basically embittered alkaloid water. The jars of waste remain sealed and refrigerated in darkness after they donated it to us until we used it for the experiments. Now, with the 3 different types of extract or effluent that we had, we intended to run 2 things. A spectral analysis to prove the qualitative presence of the alkaloids in these substances, and tests for repellency and anti-feeding tests with the beetles that we raised in the biological study. Due to COVID restrictions, it's been quite complicated. We have only been able to run alkaloid detection analysis on the aqueous effluent, but we're also carrying out the tests on hydroalcoholic extracts. We just don't have the results for those 2 yet. Sorry. Okay. So in order to explain to you a little bit about how we prepared the effluent for the alkaloid detection analysis. We used something called Dragendorf's reagent, which is a color-based reagent intended to detect alkaloids in a sample. We followed the Srividya and Mehrotra 2003 method, which you can definitely check in the references of the paper for the procedure, and which I'm only going to show you very briefly here. So we separate 10 milliliters of the effluent, add the DR, the Dragendorf reagent, leave it for 48 hours, centrifuge it, decant the precipitate, dissolve it, make a standard solution, and create different dilutions for it, which will then pass through a UV-vis spectrophotometer versus a control sample of nitric acid and thiourea. So I'm afraid I'm not a chemist myself, so you're welcome to contact my colleague Diana Parada if you'd like more details on this. But the maximum absorbances that came out in the analyses showed consistent positive ranges for alkaloids in the 280 to 325 nanometer range, which shows that the aqueous effluent, as obtained from the factory, definitely contains alkaloids from the Tawi plant. Now, having proven the presence of alkaloids in the aqueous effluent, and assuming high concentrations in the hydroalcoholic extracts, we ran biological experiments with drywood termites selected from a pinewood breeding nucleus in an unmodified environmental conditions in an enclosed and darkened space to minimize disruption. We used the Sharma and Rania methodology from 1998 on repellent and antefeedant activity and modified it slightly for the experiment. And you can also check this in our bibliography. We chose to use termites at this point to complement this study because Tricholinus ovarius, as I've mentioned, proved to have a really long cycle spanning over 2 months. And we needed to run the experiments within the time frame allowed by our grant. The drywood termites, in any case, are also a very big problem in Peru, so it was great to be able to test on them as well. This part of our research was carried out by my entomologist colleague, Eliana Quispitupac. We cut 15 9-centimeter diameter discs of number 2 Wapman filter paper and separated them into 3 groups. 2 groups received 1 milliliter of either hydroalcoholic Tawi leaf extract, the TW group, or 96-degree ethanol, the OH group. And we allowed them to dry for 48 hours. The 3rd group was not modified at all. We just used this as a control and we called it the C group. After drying, each disc was placed in a petri dish with 30 specimens of termite workers, so Cryptotermis brevis, and their behavior was observed for 30 days in a permanently darkened 1 meter cube plastic breeding chamber in the dark. And we recorded temperature and humidity throughout. So we use a formula shown here to calculate the antefeedant index, which is the rate at which the insects are consuming the treated versus the untreated papers. We weighed the papers every 3 days, for 30 days, and as you can see by the pictures, we got significant results showing that Tawi extract is more effective than pure ethanol, both of which are more effective than nothing at all at protecting the filter paper from the termite consumption over 30 days. So we intend to repeat this experiment with Tricholinus when we have more specimens, which hopefully will be over the next, well, I guess it's August now, so in August. So hopefully we'll be able to talk about that later. Repellence was evaluated based on feeding activity and mobility on paper, which had different treatments. So Tawi leaf hydroalcoholic extract or ethanol in distilled water. And this experiment was modeled on one by Rech et al. In 2015, which we will, which you can look at in our bibliography as well. The filter paper for this experiment was cut into 9-centimeter diameter discs and then in half. So some halves received 1 milliliter of treatment and were air dried for an hour, and the others were left clean as a control. So one untreated and one treated half were set side by side in different petri dishes, and we placed 30 adult worker termites in the middle of the dish. For 30 days, we counted their location every 4 days, and we repeated each experiment 5 times. We kept the dishes in a darkened breeding chamber, and we measured temperature and relative humidity throughout. Repellence percentage, or RP, was calculated using the formula shown here, where Ca, or Ca, is the number of insects present in the control area, and Ta is the number present in the treated area. We used a t-test to determine the significant difference between the treatment and control means for the RP test, just to make sure. So the RP was also carried out with 20 specimens of Trichorinus herbarius. We didn't have 30 at that point, unfortunately. And we did it with the hydroalcoholic seed extract in March and April of this year. It was not possible to increase repetitions with the beetle because of the low number of adults we had, but these are still pending, and we're going to run these tests again with the aqueous effluent as well as soon as we can. The table above applies to the repellence tests on termites. The RP in termites showed a significant difference between treatments, with a high repellence displayed by the tawi extract than by the ethanol, and you can see that it was 94.4 versus 84.4 for ethanol. The repellence tests on Trichorinus herbarius presented both greater distancing behavior and a higher index for tawi extract at 70% than for ethanol at 10% only. So, although only qualitative analysis could be done with Trichorinus because of the COVID-19 restrictions, they just really affected the number of repetitions we could do, the tawi extract is evidently capable of a meaningful repellent effect. So this is more or less what we've got so far. And thanks to the entomology team, actually, while we were running these experiments, we were also able to collect other insects along with the Mexican brook beetle, and we'll be able to produce an IPM manual and glossary for local pests in Peru. Our results so far have shown that sufficient alkaloids can be obtained from the leaves and seeds of the tawi plant to encourage significant antefeedant and repellent behavior on both termites and Mexican brook beetles. The characterization tests on the hydroalcoholic extracts and the remaining direct bioassays with the aqueous effluent are ongoing right now, and we hope to publish them within the next year. Generally speaking, the mass production of waste tawi effluent from the food industry could supply a cheap, effective, and sustainable substance for the large-scale availability of safe repellent, both at the Peruvian level and in global heritage markets. So we think that a patent in this respect could lead to the commercialization of a heritage-friendly product whose revenues could be reinvested into further research. We're also currently researching a biopolymeric substrate to hold the extract based on tawi pods, which could further enhance repellent properties. So in the pictures, you're seeing a test with pea pods, but we would do this with tawi. Resulting extracts and substracts will also be audit tested in the future to evaluate suitability for museum use, and the mode of use for the extract will be determined in the future based on future experimentation as well. So it's really promising, and we're quite excited. We would like to thank Concitec World Bank for the improvement and expansion of services in the National System of Science, Technology and Technological Innovation, because they're the ones who gave us the grant for this research project. We thank the Ministry of Culture of Peru through the Pachacamac Museum, the Museum of the Archbishopric of Lima, the Andean Baroque Route, the University of Delaware, and State University of New York at Buffalo. We also want to acknowledge the technical support of Malú Chavez, Piero Guayla, Marcelo Espinosa, and Rosana Kwan throughout these experiments. We definitely need to thank William Trujillo from Tawi Company for the donation of the tawi leaves, seeds, and effluents. And I would like to thank Tony Irwin for the initial photographic identification of Trichorinus herbarius, and Juan Carlos Rodriguez and Karina Visurraga for their administrative support. Thank you very much. Good morning.